Hello and welcome to Oregon State University's Farm to Fork webinar series. My name is Sherry Cole. I lead the Sustainable Food Manufacturing program in the Department of Food Science and Technology at OSU, and I'll be your moderator today. First, we want to thank you for taking the time to join us. We know that you have a lot going on, and I'm sure for most of you, this has been a particularly challenging week. So we hope you're all safe, and we're happy that you've taken the time to join us today. Our intention with this series is to really bring the stories of how our food systems work and how, and most importantly, those are really changing to become more sustainable, equitable, and resilient. The food system is really diverse. And so our hope is that bringing you stories that reflect that diversity is of interest and helpful to you. Before I introduce our speaker, I just want to briefly talk about some logistics. First, we really do want this to be, we're recording the session. We make that available on the website. So, please feel free to share that afterwards. We have all of the webinars have been recorded, and there's really a treasure trove of information there. The other thing is we want this to be as much of a conversation as it can be. And so we've left time at the end for your questions. So you'll see the Q&A feature at the bottom of your screen. If you click on that, you can add questions. We'd love to hear from you. The other thing you'll see is that if you see a question that you like, you can upvote it. And then those questions rise to the top of the list. I'll remind you kind of halfway into the session if you haven't asked questions. So it's my pleasure today to introduce our topic. I am really curious about this one as well. And so please join me in welcoming Dr. Chris Curtin present a mostly happily ever after story of microbial cooperation.
Thanks, Sherry, and hello to everybody. Full disclosure, I make kombucha at home. I drink kombucha and enjoy it. I'm going to talk about how this works as a fermented food system. And at the end, I'll talk a little bit about what things that you may want to know about kombucha, particularly relating to whether it's good for you. I'll clarify first by saying that we're talking about tea, like black or green tea based kombucha that's fermented with a SCOBY. I'll talk about a SCOBY in a minute. I'm not talking about the beverage that's sometimes made in Japan from kombu seaweed. So very different beverages. What you'll learn today is I'll describe how kombucha fermentation works. I'm going to talk about the microbial cooperation that is happening inside of a kombucha fermentation. And then I'm going to talk about kombucha and health. Along the way, I thought I'd throw in a few bits of Australian slang because if you didn't recognize the accent, that's where I'm from. I'm from Adelaide in South Australia. I moved to Oregon in 2016, and one of the first things you do when you move to a new country is go to the supermarket and fill up your fridge. And I saw this, and I was amazed. Well, not this shelf exactly, but walk into any supermarket in Oregon in 2016, early 2017, and you would have found a lot of kombucha. And in Australia, kombucha was just kind of something that was around the fringes, not very commonly consumed. And I'd never seen it in a supermarket and hadn't consumed kombucha. Now, some Australian slang. When I got here, I thought maybe the people who live here have a few kangaroos loose in the top paddock. Because like I said, I hadn't had kombucha, and I was aware as a microbiologist of how it was made, and that the fermentation looks something like this. It involves a mysterious thing called a SCOBY, or a symbiotic culture of bacteria and yeast. So from a do I want to consume this point of view, I looked at this and thought no. From a microbiologist point of view, I thought this is really cool and interesting, and I need to suss it out. So I started doing some research in my lab on kombucha at that time. Also, many of you would be aware that kombucha is everywhere and is a big industry, projected to continue growing over the next decade. And if I went back to 2016, there were similar slides then, projecting 20+% year-on-year growth. So growing industry and very important in Oregon. Quite a few significant kombucha producers are based here. What's driving all of this is that for a long time, and going back to when I first moved here, fermented foods had gained a lot of traction in terms of nutrition and being considered as superfoods, and kombucha is within this category. And in fact, fermented foods have been for the last 6 years the number one, um, considered the number one superfood amongst nutritionists. But as a microbiologist and a food scientist, I'm more interested in how does this actually work as a fermentation system. So I'll share some of that with you now. Oh my God, I'm so sorry. Always good when I don't share my slides. My apologies. Everybody can hopefully see my slides now. Now, the good thing is you didn't miss anything important because it was mostly just some nice visuals to make what I was saying sound more interesting. Now we're going to get really to the meat and bones of this and talk about how kombucha is made and how the fermentation system works. Right, so kombucha, the ingredients are very simple. We start with tea and sugar and water. So when I make kombucha at home, I get my ingredients together, which basically are the tea bags, some sugar, previous batch of kombucha. I'll talk a bit more about the batch component of this later. Boil some water, dissolve some sugar, steep your tea for about 10 minutes, remove the tea bags, let that cool down. That part's very important because if you've got boiling water and you're about to add that to live microbes, that doesn't work. So let it cool down. Then you add your freshly prepared sweet tea to your previous batch of kombucha that you've left a little bit of to inoculate it, and that's how you make kombucha. And the fermentation system itself, from a home fermentation point of view, is very simple and takes care of itself nicely. There are some more challenges when you scale that up to make kombucha commercially that I'll get to later. So when you make this jar, so now we've got that freshly sweetened tea. I've added that to the material that was already there, which is the SCOBY. Now it looks like this. You let that sit in a nice warm place, preferably warmer than room temperature. You're really looking for the hottest room you can find. Not as easy to make kombucha in the winter months. I tend to make it in summer when I can put my jar in the garage where it's a bit warmer. All right, you're going to leave that for 7 to 14 days. What's going on inside the jar? And this is the cool part, because you have live microbes that are carrying out their metabolic activities that enable them to grow. And what they're doing along the way is converting the sugar initially into ethanol. Now, kombucha is considered a non-alcoholic beverage when it's made correctly. So we don't actually want the ethanol, but the ethanol is important because it's used by bacteria to make acetic acid. And if you think about kombucha, it smells kind of like vinegar. And that's why, because the same bacteria that we use to make vinegar are involved in this fermentation as well. So this is all happening in the jar at the same time. And that's where it's different to vinegar, because in vinegar, we take something that's already alcoholic, like wine, we add the bacteria, and we turn it into vinegar. The magic here is that it's all happening at the same time. Now we finish that fermentation, 7 to 14 days or so, and then we'll take that liquid and transfer that into a closed bottle with some fruit purees or whatever you like to make it flavorful. At the same time, we're transferring the live microbes. If you're doing this at home, you haven't done anything to remove them. So that bottle now contains live microbes, the fermented tea, and the flavors. And what's happening inside that, or during this stage, which is called secondary fermentation, is that any of the sugars that are left over from the original fermentation, plus any that you've added in fruit purees, they're continuing to get fermented into ethanol and some other products. Mostly what we're looking for here is the carbon dioxide, the bubbles that make it fizzy. So it's a very short fermentation. We don't get much ethanol accumulation when things go well. So now we have this bottle of kombucha that still contains live microbes. And when we when we drink that, we're consuming the live microbes, and they make their way into our gut and contribute to the overall composition of microbes inside of us. So this is the health component of kombucha. Oops. All right. So I've mentioned this thing called a SCOBY, or a symbiotic culture of bacteria and yeast. And what I want to describe now is what that actually means. What is the symbiosis? Because it's really at the heart of what allows those fermentation processes to happen in the same jar, where we got ethanol forming and being turned into acids that are like vinegar. So back to this jar, where we've got live microbes and these metabolic processes happening. Oh, forgot to point out earlier as well, an important component of doing a kombucha fermentation is that initially the jar is not sealed. We just have like a cheesecloth or something else, like a coffee filter over the top, allowing oxygen in. That's important because the type of microbes that are there need that oxygen. So what's going on? What is the nature of this microbial operation? So initially, it's driven by the yeast that are present in this symbiotic culture of bacteria and yeast. They're the microbes that split the sucrose, which is what we sweeten the tea with, standard sugar. They split that sucrose into sugar molecules that can be used by other organisms. At the same time, they're fermenting those to ethanol and carbon dioxide. This is where the symbiosis comes in, because the acetic acid bacteria that we need to make the vinegary-like aromas and flavors and the acidity, they can't use the sucrose that we use to sweeten the tea with. They need the yeast to go first, break that down. And in fact, the best energy source for acetic acid bacteria in carrying out this fermentation is in fact the ethanol that the yeast are making. So it's kind of like a relay race where the yeast splitting the sugar, fermenting some of it to ethanol, and feeding the bacteria. Now, there are some other bacteria present as well. They're the bacteria that we find in fermented foods like sauerkraut, but they also don't tend to like using the sugar that's in the initially sweetened tea, so they also rely upon the yeast. So this is from a symbiosis point of view. What we're looking at here is the benefit that the bacteria are gaining from the yeast. So what do the yeast get out of the equation? Because for it to be a symbiosis, then they both need to be benefiting. And this is where I think as a fermentation system, it's really cool, because what's happening is the acetic acid bacteria, while they're making the vinegary aromas and flavors, they're also making that floaty, weird-looking SCOBY, the material that floats to the top, which is made out of cellulose. So it's a cellulose raft, essentially. And why do they do that? Because they like oxygen, and the oxygen is at the top of the jar. So they basically float themselves to the top, where they can gain access to oxygen. So what about the yeast? Because as I mentioned that for this to be a symbiosis, they have to benefit as well. The yeast that we find in kombucha actually like a little bit of oxygen as well. So they benefit from being embedded in that cellulose. And so if we zoom in on what this SCOBY looks like, we can see probably better on this side. Here's all the cellulose fibers, and they basically wrap themselves around the bacteria and yeast and protect them from the environment and give them access to oxygen. The other benefit of this is it makes, from a home production point of view, it makes carrying out a kombucha fermentation very easy, because we have this material that we can easily transfer from one batch to another. So when we finish making a jar full of kombucha and you drain most of the liquid out, you make some fresh sweetened tea. And then the way that we set that new sweetened tea to work, to carry out a new fermentation, is that we're transferring this chunky pellicle, the SCOBY at the top, and some of the liquid from the previous batch into the freshly sweetened tea. Whether you move it from one to the other, or you're adding the material back into or adding the fresh material into the jar, it doesn't really matter too much. The key process here is that we're using some of what was in the original ferment. And then the magic happens again. We get a new batch of kombucha. And you can repeat this essentially forever if you do it right. An important thing to be aware of, because the bacteria need the oxygen, the thicker this SCOBY gets at the top, it actually starts to block the oxygen. So periodically you have to peel some away and get rid of it. Give it to a friend, compost it, turn it into clothes. There are a lot of different uses for SCOBY to be sustainable. Okay, now what's happening inside the bottle? So once we've taken the kombucha and we've decanted some of that into a bottle, we've added the fruit, and I already mentioned that we have this process where we're getting the carbonation, the CO2 bubbles to make it fizzy, and that's being driven mostly by the yeast and the lactic acid bacteria, because the acetic acid bacteria can't grow in the absence of oxygen, so they stop doing anything. And all we're looking at in the secondary fermentation in the bottle is yeast and lactic acid bacteria. This is where things can go wrong, and many of you would probably be aware of a number of lawsuits that have related to alcohol levels in kombucha. Here's a few examples. So the problem here is that when you seal that jar and it contains, or seal that bottle and it contains live bacteria and live yeast, and they continue metabolizing, if you don't store that bottle of kombucha properly in the fridge, then those live yeast and bacteria are going to continue doing their thing and make more ethanol. And that's how we end up with accidentally boozy kombucha. Now, this is a problem. It's been a while since I've introduced some Australian slang. If you're not intending to be on the piss or hit the terps, or if you're a two-pot screamer, which is someone who can't really hold much alcohol, when you're drinking kombucha, two things to be aware of. One, if you make it yourself and you follow a good process and you're rigorous about it, you shouldn't really get very much alcohol built up in the bottle. It should be less than 0.5%, but it's very difficult to actually guarantee that. And a lot of home kombucha is probably going to be a little bit over 0.5% alcohol, probably more like 1% ABV. That would be fairly typical. When it's made commercially and then you buy it and it makes claims that there are claims on the bottle about being alive, be aware of that and make sure that you take that home and keep it in the fridge, particularly if you're going to give it to your kids. So just some things to be aware of. Which is a segue into kombucha and health, because if we get past that possibility of that some kombucha can be containing alcohol and are aware of that and follow good practices, then it can be a non-alcoholic beverage that for many people they consider part of a healthful diet. And this is not a new idea. It goes back thousands of years, of the idea that food can be medicine. This is something that I would consider fair dinkum. I consume a lot of fermented foods because I think there are a lot of benefits. So do a lot of other people, as I've already alluded to, in terms of fermented foods being considered superfoods. Where this can get a little confusing is when we start using terminology like probiotics. Probiotics are microorganisms that we know perform or can contribute to essential functions inside of our gut. So they're characterized to perform one of several of these functions. And there's some research that shows that diets that are rich in fermented foods can provide benefits. It doesn't necessarily mean that they contain probiotics, but they might increase the diversity of the microbes that are in your gut and contribute to some of these functionalities. So give your gut microbiome a big hug. It does a lot of good work for you. And if you want to be nice to it, you might consume some fermented foods and beverages. What I'm going to talk about is very briefly talk about is a small study we did to try and better understand what was in kombucha that we could go out to the store and purchase. Does it contain live microbes? Do they contain what we consider to be genuine probiotics that perform these known functions in the gut? So this is a study that we did a few years ago. When we looked at what was on the packages, they contain information about whether the producers perceive that they contain probiotics and whether the product has been packaged as alive. And there are often amongst these are some examples, a range of written claims on the packages that relate to the product being alive or being raw. And probiotic is a word that's used quite often. So I've already mentioned that kombucha producers sometimes get sued for levels of alcohol or sugar levels that don't match label. Sometimes they also get sued for whether or not their product actually contains probiotics, which is difficult to actually demonstrate. Um, if you consume probiotics, like if you buy a supplement, you'd be familiar with a term called CFU, or you might have seen a term called CFU and wondered what it means. This is what it means. It means that there was a microbe in that sample that could form a colony on a plate. Very basic microbiological technique where we work out how many of live microbes are there. And we can consider this a rough guide to the probiotic potential. All we're really measuring, though, is that something is alive. We don't know what is there. Unless, of course, and just to talk about our sample set, this was true for 82% of the samples. They did actually contain live microbes. So if you're going to the store and buying kombucha and you're wanting to consume it because you want to consume microbes, you would want them to be alive. So 82% of samples did meet that claim at the very least. However, what we did find in our study, and this is published, I can share a link to it for those who are interested, that samples that made claims about being alive or probiotic didn't necessarily have more CFUs. Often the CFU claims are usually made by the producer for at bottling, because that's when they last have control over their bottles or cans, is when they package it and they ship it off to the store. They might measure the number of CFUs or make a claim about that. By the time the consumer gets it, often some of those microbes have died, and the number of CFUs can be lower. And we generally observed about 100-fold less CFUs than claimed on the packages. And we analyzed what kinds of bacteria and yeast were present, and we didn't necessarily see any that were known characterized probiotics, except under the circumstances where probiotics are listed on the ingredients. I'm just labeling or showing a couple of labels here and highlighting. Now, some of these microbes, for example, Bacillus subtilis or Bacillus coagulans, they're not generally present in the SCOBY or performing the fermentation of kombucha. And for them to be listed on the ingredients, it means that the producer has to know that they're in there. And the way that they generally do that is by actually adding them as a supplement. So much like you can go to the store and buy a supplement in a capsule, kombucha producers can buy off-the-shelf supplements through a supplier, and they can add that to their kombucha to augment the number that are there. So if you're really serious about wanting to consume kombucha that definitely contains probiotics, look for that on the ingredient list. But at this point, you might feel like you might want to spit the dummy, because what I've told you is that the number of CFUs that we observe is often lower than what is claimed. And if you want to be sure that there's a probiotic in there, then you're looking for it on the ingredient list, which means that it's been added and not necessarily part of the original kombucha culture. Is that what you thought you were getting or not? That's really down to the consumer of what they're looking for. What I will finish on here is just circling back to the study I mentioned earlier that talked about fermented foods and that they can increase microbiome diversity and have health impacts that way. I personally, and the reason why I consume kombucha that I make, is that I consider that the broader category of fermented foods are helping to increase my personal microbiome diversity, and that has anecdotal long-term evidence, even if the individual microbes that are present aren't formal probiotics. I can also point you to this resource on the right, which kind of describes this difference between probiotics, which would be when you buy a supplement, probiotic or foods that contain a probiotic that's been part of the fermentation, and fermented foods as a general category. So my takeaway, other than you've learned a little bit about how kombucha fermentation works, is if you like the flavor of kombucha, then drink it for that reason. You will most of the time be ingesting live microbes. I would encourage you to take care if you are a two-pot screamer to just be aware that some kombucha can contain unwanted ethanol if they haven't been produced or maintained or looked after properly in bottle. And I'd encourage you to consume fermented foods as a general category as part of looking after your gut health. I'll finish there by thanking my lab, thanking you all for bearing with me while I totally forgot to share my screen, and look forward to answering some of the questions. Thank you.
Thanks so much, Chris, for the presentation. We have time for questions, so for those who haven't posted them in the Q&A, please go ahead and do that now. Amanda asks, what's the role of fermentation temperature? Does it just affect the fermentation rate, or are there differences in microbial populations and fermentation products?
Good question. So for kombucha, the fermentation temperature definitely affects the rate. So around 30 degrees Celsius, is it times two? No, I can't do it off the top of my head, but around 30 Celsius, you're going to get a faster fermentation. Around 20 Celsius, your kind of room temperature around now, it's going to be a lot slower. So you have to take that into consideration. It definitely changes the balance of the microbes, so the yeast to bacterial balance, and can affect the pungency, if you like, how vinegary the kombucha will smell and taste.
Thank you. Jim asks, are yeast and bacteria cultures for kombucha commercially available, like from companies that sell brewers or baker's yeast?
Not really. The challenge is the yeast is the main yeast is not one that yeast producers commonly make and sell. There are a couple, but you can get them, for example, from some homebrew suppliers like White Labs. They have yeast and bacteria that they will sell. Often, the best way to get a kombucha culture is to get the actual SCOBY, a physical SCOBY, either from somebody you know, or you can order them on Etsy and Amazon, or there are other people who sell them as well. And honestly, when I started making kombucha at home, I went to the store and bought a bottle of a kombucha that I knew contained a lot of live microbes, and made some sweet tea and dumped in that kombucha and let it sit there for a couple of weeks until a SCOBY formed. You're going to end up, what we found when we look at SCOBY from all over the country, is they generally contain the same kind of yeast and bacteria, irrespective of how the initial culture was generated.
So this is my question, just as a complement to that. So for commercial kombucha manufacturers, how are they developing the culture systems that they use?
Mostly they start out with a SCOBY, however they got it, and then they essentially train it to their system by doing these repeated batches. And if you recall, I mentioned that periodically you have to peel some of the SCOBY off, otherwise it gets too thick, blocks the oxygen, and just becomes difficult to work with. I mean, and again, thinking about the bigger the tank gets, the bigger this chunk of cellulose gets. It gets quite heavy, difficult to work with. When they peel that layer off, they put that into something that they will then call a SCOBY hotel, with some of the fermented kombucha liquid as well. And you can keep a SCOBY hotel with multiple backup SCOBYs for several months if it's well maintained. So they tend to build up their culture and then keep it as a backup, and they'll just keep rotating through their SCOBYs.
Katya asks, what are the biggest mistakes that a home brewer or fermenter could make?
Mostly being inconsistent. So if you're not consistently using the same amount of tea and sugar, and you're not performing this in a relatively consistent temperature for the same duration every time, then it's going to be all over the place in terms of the flavor profile. Some batches will be funkier, some will be cleaner, some will be more or less acidic, and you really want to try and dial that in and be consistent. But it's fairly forgiving. I mean, I have had some batches where I forget about them for 3 weeks, and really it just comes down to how you balance that out when you're blending it with different ingredients to make it taste good. Um, it's a fairly forgiving system.
Amy asks, could improperly produced kombucha make you ill?
Okay, good question there. There is some evidence. This is more of an overconsumption issue as well, though. The acidity, I mean, if you're making kombucha at home and you make it very acidic and then you consume a lot of it, then you're going to be ingesting a lot more acids than you would normally have in your diet. And there are examples of that being detrimental to health for sure. So there's some FDA guidance on that if you're interested in looking that up.
Is
The
there a
alcohol
standard of
part is the other
identity?
part as well. Um, Obviously, if you're not making kombucha quite right and you're getting too much alcohol, then that can be an issue. But if you look at the guidance on how much kombucha to drink, you shouldn't be drinking that much anyway.
Sorry to interrupt you. Is there a standard of identity for kombucha?
There's a voluntary code of, not standard of identity, but code of production conduct, if you like. There's a organization called KBI, Kombucha Brewers International. They have within their materials a standard of identity for what would be considered traditional kombucha, but it's voluntary as to whether people actually comply with that and whether they label their kombucha as traditional kombucha, or kombucha-like product. Yeah, no rigorous standard of identity.
Also asks, it seems that commercial kombucha is more fizzy than my home-brewed version. Why so?
Yeah, sometimes they force carbonate, so they'll use carbon dioxide gas to boost up the bubbles and make it consistent. When you're doing it at home, you're relying upon whatever's still alive in the bottle to continue to ferment and make some bubbles. And I've found that when I make it, as long as I put it in the fridge and then leave it for a while, then it'll get there eventually. You don't want to leave it out on the bench out on your kitchen bench for too long because you risk getting too much ethanol. But the reality is, by the time you're getting a good, if you're just letting some of the gas out just to check, and you're doing that periodically and not forgetting about it for a couple of weeks, you're probably not going to hit that point because by the time you get a good fizz, you don't have very much ethanol at all. Now, if you leave it out on your kitchen bench for 2 weeks and forget about it, it's going to be very, very fizzy and potentially explosive. It could also contain quite a lot of more ethanol than you want. So, yeah, be wary of that.
Ellie asks, how do you know when to remove some of your SCOBY if it gets too thick or by the color or something else?
I judge it by how floaty it is. So as soon as you're starting to get the SCOBY not being covered with liquid, then you have too much. So once it's starting to kind of bob and sit at the top and the surface is exposed, start ripping off at that point. You really want it to stay mostly submerged so that I did have a slide that I think I got rid of at the last minute. Something that can go wrong is if that SCOBY is exposed for too long repeatedly, then you might get mold growth on the surface, and you don't want that.
Rico asks, is there any evidence about the production of any antibiotic during fermentation?
Good question. Not that I'm aware of. The microbes that we find in kombucha, I don't think so. Like I'm not aware of any products of those microbes that would kill other microbes that we would be concerned about.
Lisa asks, I have an abundance of SCOBY. Are there any health benefits of eating the SCOBY?
I don't think so, but honestly, I haven't looked into that one very much. I know chickens like it.
Jim asks, is cooling needed to keep the fermentation from getting too warm?
Good question. Generally not, unless if you're getting above 30 Celsius, you do start to run the risk of it not functioning properly, like being out of balance in terms of the yeast and bacteria. So I don't cool mine, but I also don't rigorously monitor the temperature in my garage when I'm carrying out the fermentations. It it works pretty well on a small scale. As when you're getting larger, um, even then, it, if we compare kombucha fermentation to some other kinds of fermentations like beer or wine, where there's a lot more alcohol being made, the number of the the number of microbes that are present is much lower because the tea. I mean, you think about all we're adding is sugar and tea, and microbes like plants. They need all the different aspects of their food, right? They need some carbon, which in this case we're supplying in the form of sugar. They also need nitrogen to make cells, so to make more protein and make more copies of themselves. There's very little nitrogen in tea, so we don't get a large number of cells, so we don't get a lot of heat production at a small to moderate scale.
Rita asks, is this fermentation method like many others where your nose will tell you if something's gone wrong? Or will it
Yeah.
show up in the color of the SCOBY?
More so you'll probably pick it with your nose before noticing a dramatic change in the SCOBY color. Um, we got samples of how many in the end? 108 SCOBY from all across the country. When we put them all out next to each other, they all look different. They're all different colors. Some look, objectively quite funky to look at. You're like, wow, I'm not sure I want to consume what was made with that, but I know better because then we looked at what's actually in them. And even though they look very different, they actually still contain the same kind of microbes. And the visual look of them is more a function of the kind of tea that's used and the conditions under which they're fermented, not necessarily something to be too worried about. But the smell, yes, you'll notice if it's kind of not smelling right.
Jim asks, there are sugar-free kombuchas on the market. How is the sugar removed, and does that affect the quality of the kombucha and its health benefits?
Good question. So, um, in the pursuit of trying to have kombucha that is less risky from a alcohol point of view, some producers are carrying out their fermentations to try and get rid of all the sugar that they initially add. So all the sucrose is all being used up, and then they'll sweeten using non, not necessarily artificial sweeteners, but sweeteners that can't be fermented by the yeast or bacteria. Jackfruit is an example of a sweetener that you might find, or erythritol. So the upside of that is those kombuchas are not going to develop ethanol, so you don't have to worry about that component. There's no real impact on the what's alive in the kombucha as long as it's still consumed relatively fresh. From an overall health point of view, I don't think it has a negative impact on your gut microbiome in the amounts that you should be consuming kombucha, given that, again, you shouldn't be consuming heaps of that. And those sweeteners, again, they're not going to be metabolized by microbes in your gut, to my knowledge.
Is there Betty asks, is there a conventional ratio of tea to sugar for kombucha?
No, there's quite a bit of variation and personal practice in terms of what people do. The range of sugar concentrations that I've seen might be from typically from say 50 to 100 grams per liter. 100 grams per liter is what I've used at home. And the amount of tea, I feel like it's typically between 1% and 2% by weight. Trying to think when I make I think in gallons, I guess so, for kombucha. If I'm making a gallon of kombucha, I'm trying to remember how many tea bags, probably 10, I think, but I don't even know if that falls into the correct number, correct by weight volume. That's just what I ended up using. So there's a fair bit of flexibility there. You probably wouldn't want to go above 100 grams per liter of sugar because the fermentation will take a long time, likely get stuck. If you are on the lower side with the amount of sugar, then you're probably more likely to get a initially less sweet finished batch that you can then add sugar when you're adding fruit flavors and things like that to balance it up.
Nellie asks, Nellie asks, how is the SCOBY different from mother of vinegar?
Mostly the presence of yeast. So the mother of vinegar will be just the bacteria, and the bacterial species that we find in mother of vinegar and we find in kombucha, there is a decent amount of overlap of the same kind of organisms. But yeah, the difference is just that in kombucha you've got yeast present as well.
Air asks, oh, sorry.
But you could you could throw some mother of vinegar into to start a batch of kombucha, and the yeast will find their way in from the environment slowly, but they'll get there. So you could start with mother of vinegar as an option as well.
Claire asks, what is the effective fermentative difference between the top, middle, and bottom SCOBYs? Which do you take away?
Oh, the bottom is where the fresh growth is. So you want to take away the top. Keep the freshest stuff. Well, personally, that's what I've done at home. Is there's a zone, I can't remember exactly how far in, where it's been shown that that's where the new material is forming. But they're kind of, sometimes it isn't immediately obvious. But after a few batches, you can start to see the lines in the SCOBY. Yeah, I'll peel from the top, put that into a hotel if you want to keep it.
Time for one more questions. I love all these questions. Thank you, everyone. Katya asks, does the type of fruit juice used in the second fermentation significantly affect the development of fizz or success of the yeast and bacteria?
Yeah, it will, because as well as adding sugar, we're also adding nutrients that might increase the number of yeast and bacteria that will grow. So just adding sugar, we're not adding any nutrients. Whereas if you add a berry juice, for example, that will introduce some other nutrients. So berries are an example that they might make the end products slightly funkier, or can do, because they introduce other compounds that the yeast and bacteria might metabolize and convert into other kinds of compounds that are aromatically active.
Thank you. Well, thank you so much, Chris, for a fascinating presentation, and thank you everybody for all of your questions. I apologize that we couldn't get to them all. You'll see that, so we would love your feedback. These are only as useful as are of interest to you. So you'll see a link in the chat for a short survey. Please take the time and to give us your feedback. We're also doing these monthly. So our next one will be Friday, February 16th, and the topic will be what can novel non-thermal technologies do for sustainable food processing. So we wish you all a great weekend, and thank you again for joining us, and we hope to see you next time.